Okay, so we are going to study about today is about the topic gametogenesis. Okay, what is gametogenesis? Gamete means what? Gamete. Okay, gamete genesis. So here the gametes are male gamete and female gamete. Male gametes are sperm. Female gamete is ovum. Right. So we are going to study in the topic of gametogenesis is about spermatogenesis and oogenesis. Spermatogenesis is called as spermatogenesis. Okay, and ovum production is called as oogenesis. Correct. Let us understand details regarding individual. Before going to the individual, what is spermatogenesis and oogenesis, let us understand in general what is gametogenesis process. Okay. First we'll see the background behind the gametogenesis is about where do they start their production first. Okay, so they are called as before the sperm cells or ooh cells, either sex, they are called as initially called as a primordial germ cell. Okay, they are called as primordial germ cell. It's called as PGC. Okay, these primordial germ cells will give rise in male as a sperms and in female are ovum. They first start their production. See, if it is a developing embryo, okay, this is embryo, this is the head, this is the head and all. This is the yolk sac. Okay, this is yolk sac. This is the wall of the yolk sac. They first start production on the wall. Okay, these are the PGC first start to produce on extra embryonic mesoderm of yolk sac on the 4th week of intrauterine life. On the 4th week of intrauterine life. So what we understood this one. So they gives rise to male and in female as sperm and ovum. They resides. Where do they resides in? They resides in extra embryonic membrane, not mesoderm. Okay, membrane, extra embryonic membrane of the yolk sac. Correct. The yolk sac, and production will be on 4th week. Right. This is on the 4th week. Right. See, you already know about this disc, the production of the disc, no, bilaminar disc, trilaminar disc, like what you call germ layers, right? So before it was bilaminar. This is actually bilaminar disc. The above it is epiblast and below it is called as hypoblast. Together they are known as bilaminar disc. Okay, bilaminar disc. See, among these two, this is epiblast. From this epiblast layer, these PGCs are going to form. Later this epiblast is going to convert. After this here, there is a mesoderm is going to come in this area. Later mesoderm is going to come here. After this mesoderm formation, the name of epiblast will change and we call it is epiderm is called as endoderm and hypoblast is called as ectoderm. Okay, so PGCs are going to form the epiblast. Indirectly, we are going to say that endoderm is the origin for the PGCs. So what we say is about they they are derived from epiblast. In bracket we'll write endoderm. Correct. Right. Now we'll see migration. They are not going to stay there in the yolk sac only right now. So this is a baby, right? And you know this, if it is a female baby, suppose here it is its ovary. Okay, there are the PGCs are forming here, cytodifferentiation taken place. Okay, from here they have to migrate towards the ovary, and while going there it has to cross the gut. It has to cross the gut and mesentery. It has to cross gut mesentery and will come here. When they come here, from here they will come here crossing the gut mesentery. When they come and reside on the surface of the ovary. Okay, we'll draw this separately. This is ovary and these are the PGCs will come and locate on the surface of ovary, and they are covered by a particular cells which are known as coelomic epithelium. They are going to covered by a special type of epithelial cells which are known as coelomic epithelium, or we can call these are the coelomic cells of the epithelium, right? They are going to covered by coelomic epithelium here. And how do they move from here to here? They move in a motion called a amoeboid movement. What is a movement? Amoeboid movement will be there, right? So what we write? We write here as they migrates from where? From yolk sac, wall of the yolk sac, right? From yolk sac to where? To gonads. To gonads no. So gonad by which type of movement? Amoeboid movement. Okay, via it they crosses what? They crosses gut mesentery, right? This is the point. Then in what we will write? We will write PGCs after moving on the surface of the ovary. PGCs are invaded by cells of what epithelium? Coelomic epithelium or coelomic cells, correct? So this is about the background, how they form, how they migrate, how they locate on the ovary, when they migrate. Okay, let us discuss regarding about gametogenesis. Let us discuss about gametogenesis now. So what is a gametogenesis? It's a process of formation of gametes from germ cells, right? It is a process of formation of gametes from germ cells, right? In male already you know, tell me, in male PGCs, okay, PGC remain what? Remain dormant. You know this one, okay? In men, the PGCs are going to remain as a dormant from 6th week or 4th week. When they will come up to 6th week, it will reaches to the testis in male and in ovary to the female, and they remain in male as a dormant. Dormant means they struck their movement there, means their division movements, cell division movements will be stuck there till puberty. Then the seminiferous tubules undergo maturation. Then the meiosis process forms the spermatogenesis. So there is a difference in men and women. Okay, in male what happened? The PGCs remain dormant. From which week? From 6th week. Okay, 4th week they start formation. 6th week they reaches there. Okay, and they will become dormant till when? Till puberty. Till puberty they will remain dormant. Then what will do? Then seminiferous tubules. Okay, seminiferous tubules undergo what? They will go undergo maturation. Undergo maturation. Then what will happen? The meiosis. Avag start agtao. Meiosis process forms what? Spermatogenesis. This is what the thing in men. What about in female? In females the case is different. So in females what happen? In females, okay, in females PGCs are going to get differentiated to form oogonia first in the 5th month. Okay, in the 5th month they will going to form 4th week they are start migrating. 6th day reaches ovary. Then up to 5th week, up to 5th month, the meiosis process already starts, and they form primary oocyte also under the meiosis, and get arrested in the diplotin stage of the prophase. Okay, then again meiosis 1 will complete, but meiosis 2 will not continue, and they will get arrested up to the puberty. See, from the in the intrauterine life, 5th month only they started their meiosis process. In the 5th month they stop, and the baby is going to born, and the baby will get attained to puberty. Then again the continuation process will start about that meiosis process, and each month, every month, they among 10 to 15 or 10 to 12 primary oocytes, among 10 to 12 primary oocytes, one will complete the meiosis and to form the secondary oocyte to get ovulated. Until then they are get arrested. Meiosis process started in the intrauterine life, but it will get arrested in the intrauterine life only. Then it going to stay in the pause condition. Later it going to complete after the puberty. Not all oocytes are going to convert them into the secondary oocyte. Every month 10 to 15 oocytes are going to get maturated. They are selected for the every cycle for the continuation of the meiosis process. They will get the chance. Among this 10 to 15, one is going to get maturation. The rest of the cells will get degenerates. Okay, so what we write in female, PGC differentiates. differentiates to oogonia. Okay, the oogonia in fifth month. In fifth month, and what they form? They form primary oocyte. Okay, they form primary oocyte. They enter which process? They enter meiosis process. Okay, meiosis process, and get again arrested. Get arrested in prophase. Which phase? In prophase. Again at puberty, right? At the puberty, each month how many? 10 to 12 or 10 to 15? 10 to 12 primary oocyte. Okay, I will discuss about what is primary oocyte, what is secondary oocyte later, but you should know this concept first. Primary oocytes, among these 10 to 12, one will complete what? Meiosis process. See, that is true, not meiosis 1 process. If I specifically speak, meiosis 2 will not get complete. Okay, regarding that we will discuss in some other class why and in which phase and all. Okay, meiosis 1 will get complete to form secondary oocyte. Only secondary oocyte. Okay, and get ovulated. See, when the ovulation happens in the puberty, the meiosis process is not completely complete. Meiosis process is complete. Full meiosis process is not completed yet. Okay, it's still in a secondary oocyte stage. Done. So when it is going to complete, the meiosis 2 is when it is going to complete, it's only fertilization happens. Then the meiosis 2 will complete, or else they die like that only. Okay, so only at fertilization stage, that too in fertilization stage only they are complete. Okay, meiosis 2 will complete. Okay, so this is about general form of oogenesis. Detail we'll study in next class, but this is general concept of oogenesis. Okay, so let us study first about spermatogenesis. Okay, we will see about spermatogenesis. You know about the spermatogenesis, right? What is the definition of it? Define spermatogenesis now. It is a process. Okay, spermatogenesis is a process of formation of sperm. Formation of sperm from what? From spermatogonia. These are nothing but PGCs, right? Spermatogonia. In which part of the testis? In the seminiferous tubules of the testis. So what is the definition? It is a process. It is a process of formation of sperm from what? From spermatogonia. Where? In seminiferous tubules of testis. Okay, so already you know what is the site for the spermatogenesis. It is the seminiferous tubules of testis. Seminiferous tubules of testis. Okay, what time it will take place? When? Okay, what is the time? Time is after puberty. In male, no, it is after puberty. After puberty till old age. This cannot be said which age is old age. After 70, some people may still have the spermatogenesis process still going on in the 80s also. So age we cannot say when it is going to stop exactly. But in old days, gradually they are going to stop their production. So duration. What is the duration of production of the one spermatozoa from spermatogonia? It will take up to 64 to 74 days. Okay, 64 to 74 days. We will divide this one as per the stages. Okay. Then what is a responsible hormone? Which hormone is responsible? As you generally know, it is a testosterone. Testosterone is a responsible hormone, right? Now we'll see the process. See, the process of formation of spermatogenesis we can divide into 3 steps. 3 steps we can make. So first step we call it is a spermatocytosis. Okay, first step is a spermatocytosis, which approximately takes 16 days to complete. Then the meiosis process will start. See, in the spermatocytosis there is a mitosis process is there. Okay, spermatogonia is going to form the spermatocyte. Okay, so there the germ cell is going to become a sperm cell. Okay, that duration is actually the mitosis is going to take place in 16 days, right? Among the end product of spermatocytosis undergo meiosis process to form spermatids. Okay, spermiogenesis, spermatogonia to spermatid condition is a meiosis process where it will take around 24 days. Later this spermatids will convert into spermatogenesis, spermiogenesis. They are not in the shape of sperma till spermatid. Okay, the cell modification takes place. The certain changes will happen in the cell morphology and they attain the shape of the sperm, and this process is called as a spermiogenesis. Again it will take 24 days. So total 64 days it will take. Okay, so approximately first step is spermatocytosis. Site. Okay, spermatocyte formation. It will take approximately 16 days. And the second step is meiosis process. Okay, this meiosis process will take up to 24 days. And the third step is spermiogenesis. Spermiogenesis. Again it will take approximately 24 days. Total 64 days period is there. Okay, we can do this thing. Right, now first step, second step and third step. Let us discuss about one by one. So we will take first step, spermatocytosis. What happens in spermatocytosis? See, here the spermatogonia will convert into primary spermatocyte mainly. Okay, when the spermatogonia is there, no PGC, that will convert into primary spermatocyte. Further primary spermatocyte undergo meiosis process to form secondary spermatocyte. Then it will convert to spermatid. Okay, so now it is a process here. The spermatogonia is going to form primary spermatocyte. This process is spermatocytosis. So what happens in the primordial germ cell is there, no? On the primordial germ cell, testosterone will come and act a role here, and the formation is a spermatogonia. Before it is a just a PGC, sperm cell, PGC, primordial germ cell. Testosterone will come and act on that, and that will convert into spermatogonia. Type A, there are type A and type B. Again in type A dark and type B dark is there. We'll discuss about it. No, don't worry. So first point what we write is spermatogonia to spermatogonia to primary spermatocyte formation. This is a spermatocytosis. So what happens here? This primordial germ cell, primordial germ cell, which we call as PGC. Okay, on that testosterone will act. Testosterone is going to act and going to form spermatogonia. Okay, which is called type A dark, right? Type A dark, type A dark variety of spermatogonia. This will again go mitosis. See, this spermatocytosis always have a mitosis process. Okay, mitosis. Again, when mitosis are going to form, there are two cells, right? What will happen? Spermatogonia type A pale is going to form. Okay, spermatogonia type A pale, spermatogonia type A dark. Okay, so this is the thing will happen. Pale cell will undergo again mitosis. Okay, this spermatogonia type A pale cell will undergo mitosis again to form spermatogonia type B. Okay, spermatogonia type B cell. This type B cell will undergo meiosis process, right? So this dark is going to form the pale, then pale is going to divide to type B. This here also type B, same cell. Okay, this spermatogonia type B cell will undergo again mitosis. So what is going to come here? The primary spermatocyte is going to form. Primary spermatocyte is going to form. Okay, till here. After this, meiosis process will start, right? Till here, from the primary spermatocyte till the from the spermatogonia till its formation of primary spermatocyte, this whole process is called as spermatocytosis. This is called as spermatocytosis, which will take 16 days, which we have discussed in short, right? After this, whatever is going on, it's about the second step, meiosis process. Okay, now it is about meiosis process. Let us see what is going to happen in meiosis. You already know that meiotic division is having two types, first meiosis and second meiosis. See, any cell will undergo meiosis, will form primary will become meiosis 1, form second. This is a primary, this is secondary. This is meiosis 1. Okay, secondary is going to again undergo reduction division, then again daughter cells are going to form, and this is the gamete. Okay, this is meiosis 2, right? So usually the meiosis will have two meiosis stages are there. Okay, so let us see one by one here. So what is going to happen here? This spermatocyte already have how many chromosomes? 44 XY. We are speaking about spermatocyte, so we will take XY. If we were speaking about oogenesis, we would have spoken about 44 XX, right? So what happens in meiosis process will continue from here only, right? Primary oocyte, which is having 44 XY. So first meiosis process, meiosis 1 is going to happen, reduction of the chromosomes but not the DNA. We will discuss about this detailedly later. Okay, then it is going to form secondary spermatocyte, okay, which is having how many chromosomes? 23X or 23Y, either, right? Half number of the chromosomes happen. Next, what happened? Meiosis 2 is going to happen. Then the spermatids are going to form. Here also they have 23X or 23Y chromosomes are there, right? Then after the spermatid, okay, after the spermatid, the third process is going to come here. This third is a spermiogenesis. What is spermiogenesis? Spermiogenesis is what? Spermiogenesis is a process of metamorphosis of the spermatids by which they converted into spermatozoon. See, the cell is like this here. When it will come, it is a more round like a normal cell having a nucleus inside. When they will become sperm, they will have the head like this, middle piece like this, and tail like this. Okay, so this is the conversion from the normal cell to this type of cell. So this process of metamorphosis of this spermatocyte to the sperm cell, this process is called as spermiogenesis. See, there is a difference between the spermiogenesis and spermatogenesis. Okay, you should know the difference. Spermatogenesis is a whole process from the primary spermatocyte to sperm, complete process, right? But spermiogenesis is the third stage of the meiosis. What happens here? Spermatids, okay, we'll write the notes also for your purpose. Process of metamorphosis. What is metamorphosis? Change in the structure of the cell. Okay, shape of the cell, not structure, shape of the cell. Metamorphosis of spermatids. Okay, by which spermatids they convert. Okay, they are going to convert it into what? Into spermatozoon or spermatozoa. Both are same. Okay, what is the process? See, what is going to happen in this process? The process involves nucleus condenses. First, what happens? See, this is the spermatid and this is the nucleus. For suppose this is a nucleus. Okay, Later what happens, this nucleus will come here, condenses and come at one end. Okay, see, this is a cell now. It will going to come at one end. Later what will happen, the cell is going to come form like this, and nucleus is going to come here in this area. Okay, here will come, and this is a neck, this is again middle piece. Okay, we will discuss about it later, but here the nucleus will get condensation, and this is a head portion, right? So nucleus from this to this form and this to this form will going to convert, and this process is there, no? So what will write, the nucleus condenses, and moves towards where? Head, right? Head. Okay, we can call the one pole now. Okay, one pole of the cell. Right? Now, this cell not only have only nucleus, no, it does have some Golgi apparatus here. We can make it is we'll took a different color for this one. Okay, this is a spermatid, this is a nucleus. Okay, we'll mark it as nucleus. Okay, now we'll say this red color as an example. Okay, not exactly, these we will name as a Golgi apparatus. So what they will come, they are going to come in front of the nucleus here at that pole, and they are going to convert as one special type of cap around the sperm here, and this special type of cap is called as a acrosomal cap, because it's having acrosomal cap, it contains acrosane mainly as a chemical substance called acrosomal, but made up of Golgi apparatus. So what we'll write, we write next process, this is a process number 1. Second process is Golgi apparatus. What they do, forms acrosomal cap. Going to form the acrosomal cap. Okay, again we'll take a cell here. This is a cell. Okay, this is a nucleus, right? This is going to form the shape of the sperm. Okay, and this is a tail. Now here our nucleus came, condensed the form in the head, one pole of the cell. Then again it's going to form the nucleus form, and we had a Golgi apparatus here. They are going to come and form the acrosomal cap. This is a acrosomal cap. This is Golgi apparatus, right? Okay, now the cell do have centrioles. This is what? Centriole. Okay, so these centrioles are going to divide into, like, we will take another phase of the cell here in between, converting cell of spermatozoa. Okay, this is a nucleus, suppose it's a middle stage, and we have red as Golgi apparatus will come here. Later they are going to form the acrosomal cap. So we have this one as centrioles. This centriole will become like this. So this is near to the nucleus, we call it is a proximal centriole. Okay, and this we call it is a distal centriole. Okay, see proximal centriole, this one will come and locate in the neck portion here. Okay, and it starts producing axial filament. We call it is a axial filament. Okay, and this is actually the proximal centriole, subproximal centriole, and this one will come here between the tail and the middle piece and form this ring-like structure. Okay, ring-like structure. Can you see this one? And this is the ring-like structure called as annulus. What it's called as annulus, through which the axillary filament passes to the tail part. Okay, so what we write, this is a third point what we write. Okay, centromere divides into proximal centriole and distal centrioles. Okay, centrioles divides into A proximal centriole and B distal centriole. Correct? Next, what has happened? Proximal centriole comes to lie in neck of sperm or spermatozoa and gives rise to what structure will gives rise to? Gives rise to axial filament. Okay, axial filament. Next, so next B, what? Distal centriole. What it will do? Distal centriole comes to where? Okay, comes to lie in which part? Middle piece and tail piece. Okay, in between middle piece and tail as what it is called as annulus ring. Correct? Annulus ring. So next, what happened? See what happens here, I will tell you. As this process is going on, when this process is going on from here in here, there is a cytoplasm, no? Cytoplasm is there in that cytoplasm, and see, when you stitch your cloth is something different before. When tailor do the stitch your cloth to form a shirt. Okay, so while doing that shirt, he will cut the cloth to stitch, right? As they will remove, they will form the shirt shape, no, like this. Here the tailor is a biomechanism which is doing this shape, from this shape to this shape, where the cytoplasm will also get rid of, along with the cell membranes also get trimmed off. Okay, so they will remain as a residual bodies, and these residual bodies are called as Reynolds. Okay, Reynolds residual body. Okay, so we write the next fourth step, right? Reynolds Reynolds residual bodies shed off. Okay. Then what is the fifth process you name? Fifth process. See, suppose you take, this is the seminiferous tubule and this is a lumen of the seminiferous tubule. This is very tiny. I am making it a big area. Somewhere here on the wall of the seminiferous tubule, these are the spermatogonia. Okay, these are the spermatogonia, right? They undergo process of mitosis and meiosis. All these things are going to happen, and completely when they will come here, they will be converted into spermatids in this area. Okay, so once they form, metamorphosis happens, and here the shape of the sperm will come, and later they release into the lumen, like here they released into lumen. This releasing of the completely formed spermatozoa to the lumen is called as a spermiation. Okay, so releasing of the spermatozoa will happen here. So releasing of spermatozoa or spermatozoon to the seminiferous tubule. What it is called as spermiation, right? There are three words will come: spermatogenesis, spermiogenesis and spermiation. All are different. Okay, so this is a general process of spermatogenesis.